Hello students, I'll talk about the structure of the immunoglobulins today. Introduction. Antibodies are the antigen-binding glycoproteins that are synthesized exclusively by the B cells. Each glycoprotein has different amino acid sequence and different antigen-binding sites. They are collectively known as immunoglobulins or IG and are the most abundant protein components of the blood plasma, constituting 20% of the total protein components. Basic structure of immunoglobulins. Antibody molecules have a simplest structure of Y shape with two identical antigen binding sites. Each of them are at the tip of the Y arm. Therefore, they are described as bivalent. Antibodies have a common heterodimer Y-shaped structure made up of 4 polypeptide chains. This heterodimer structure consists of two identical light chains of about 220 amino acids and 25,000 molecular weight and two identical heavy chains, each of about 440 amino acids residues having 50,000 molecular weight. Each light chain is bound to a heavy chain by a disulfide bond and other non-covalent interactions. Thus, antibody is a heterodimer of heavy and light chains. Similar non-covalent interactions and disulfide bridges again link the two identical combinations of heavy and light chains to each other to form the basic 4-chain structure of antibody, which is a dimer of the heterodimers. The exact number and precise position of these interchain disulfide bonds differ among different classes and subclasses of antibody. Sequencing of immunoglobulin. Sequencing analysis of immunoglobulins become feasible with the discovery of multiple myeloma, a cancer of the antibody-producing plasma cells. The plasma cells in a normal individual are end-stage cells that secrete a single molecular species of antibody for a limited period of time and then die. In contrast, clones of plasma cells in an individual with multiple myeloma has escaped normal controls on their lifespan and proliferation and are not end-stage cells. Rather, they divide over and over in an unregulated way. Though myeloma cells has been transformed, its protein synthesizing machinery and secretory functions are not altered. Thus, the cell continue to secrete molecularly homogeneous antibodies. This antibody is indistinguishable from the normal antibody molecules, but it's called myeloma proteins to denote its source. In a patient afflicted with multiple myeloma, myeloma protein can account for 95% of the serum immunoglobulin. In most patients, the myeloma cell also secrete excessive amount of light chains. These excess light chains were first discovered in the urine of myeloma patients and were named Ben-Jones protein for their discoverer. When the amino acid sequence of several Ben-Jones protein or light chains from different individuals were compared, a striking pattern emerged. The amino terminal half of the chain, consisting of 100 to 110 amino acids, was found to vary among different Ben-Jones proteins. These regions were called the variable or V region. The carboxyl terminal half of the molecule, called the constant C region, had two basic amino acid sequences. Both light and heavy chains have a variable sequence at the N-terminal region but have a constant sequence at their C-terminal end. These segments of highly variable sequences are called V regions, VL in light and VH in heavy chains. The constant region of light chain CL has same number of amino acids to that of VL, but the heavy chain constant region or CH has 3 to 4 times longer 330 or 440 amino acids depending on the class of the antibody molecule. The diversity in the variable region of both light and heavy chains can be traced by the differences fall within areas about 10 amino acid residues of the B region called the complementarity determining regions or CDRs. These CDRs constitute the antigen binding site of the antibody molecule. The remaining part of the variable regions known as framework regions are relatively constant. Proceeding from the amino terminus of either the VL or the VH, there are CDR1, CDR2 and CDR3. Among them, CDR3 is the most variable CDRs. Classes of immunoglobulins. All species have two major classes of light chains, kappa and lambda. Any individual of a species produces both types of light chains. However, the light chains are always either both kappa or both lambda, never one of each in an IgG molecule. The IgG of all species have 5 different types of heavy chains: gamma, alpha, mu, epsilon and delta called isotypes. The heavy chain of a given antibody determines the class of that antibody. IgG for gamma, IgA for alpha, IgM for mu, IgE for epsilon and IgD for delta. Each class can have either kappa or lambda light chains. Any individual of a species produces all heavy chains, but in any one antibody molecule, both heavy chains are identical. Thus, an antibody molecule of the IgG class could have the structure either kappa 2, gamma 2 or lambda 2, gamma 2 with 2 identical kappa or lambda light chains and 2 identical gamma heavy chains. Further classification of the heavy chains into subclasses was made by the minor differences in the amino acid sequence of the gamma and alpha heavy chains. So there are 4 subclasses of gamma heavy chains: gamma 1, gamma 2, gamma 3 and gamma 4, and 2 subclasses of alpha heavy chains, alpha 1 and alpha 2 in the humans. Antibodies are glycoproteins and the sites of attachment for carbohydrates are restricted to the constant region. The role played by glycosylation of antibodies is not fully understood, but it probably increases the solubility of the molecule. Inappropriate glycosylation or its absence affect the rate at which antibodies are cleared from the serum and decreases the efficiency of interaction between antibody and the complement system and between antibody and the Fc receptors. Chemical and enzymatic deduction of antibody structure. Porter and Edelman elucidated the basic structure of IgG molecules. Porter found that the proteolytic treatment with papain split the Ig molecule, which is 150,000 molecular weight, into 3 fragments, 2 of which were identical fragments, each with a molecular weight of 45,000, called FAB fragments or fragment antigen binding, because they have antigen binding activity. The third fragment of 50,000 molecular weight was found to crystallize during cold storage. So it is called the Fc fragment, fragment crystallizable, and it has no antigen binding activity. Edelman reduced the IZ molecule into 4 chains, 2 identical chains having 53,000 molecular weight and 2 others of about 22,000 molecular weight, each by treatment with mercaptoethanol. The last molecules were designated as heavy chains and the smaller one as light chains. Further, Nisonov uses pepsin to digest the IZ molecule and generated a single 100,000 molecular weight fragment composed of 2 FAB-like fragments designated the FAB'2 fragment. FAB'2 can bind antigen. The FC fragment was not recovered from pepsin digestion because it has been digested into multiple fragments. Immunoglobulin fine structure. The structure of the immunoglobulin molecule is organized into the primary, secondary, tertiary, and quaternary structures. The amino acid sequence of the primary structure accounts for the variable and the constant regions of the light and the heavy chains. The secondary structure is formed by folding of the primary structure back and forth upon itself into an antiparallel beta-pleated sheets. The chains are then folded into a tertiary structure of the functional compact globular domains, which are connected to neighboring domains by continuation of the polypeptide chain that lie outside the beta-pleated sheets. And finally, the globular domains of the adjacent heavy and light polypeptide chains interact in the quaternary structure, forming functional domains that enable the molecule to specifically bind antigen and at the same time perform a number of biological effector functions. Immunoglobulin domains. Both light and heavy chains are made up of repeating homologous segments of about 110 amino acid residues, each containing an interchain disulfide bond. These repeating segments fold to form compact functional units of about 60 amino acids called immunoglobulin or IZ domains. A light chain contains one variable VL domain and one constant CL domain. Similarly, a heavy chain consists of one variable VH domain and either 3 or 4 constant CH domains, depending upon the antibody class. Most heavy chains have 3 constant CH1, CH2 and CH3 domains. But those of IgM and IgG antibodies have 4 CH1, CH2, CH3 and CH4 domains. VL and CL domains of light chains pair with the VH and CH1 domain of the heavy chain to form the antigen binding site. Hinge region. The gamma, delta and alpha heavy chains contain an extended peptide sequence between the CH1 and CH2 domains called the hinge region that has no homology with the other domains. The hinge region is rich in proline and cysteine residues and is flexible that can move from 60 to 180 degrees. Only IgG, IgD and IgA have the hinge region that makes them flexible between two FAB arms of the Y-shaped antibody molecule. The large number of proline residue in the hinge region make it susceptible to cleavage by proteolytic enzymes such as papain or pepsin. The cysteine residues form interchain disulfide bonds that hold the two heavy chains together. The number of these interchain disulfide bonds varies with the variation in length of the hinge region, that is from 10 to more than 60 amino acid residues in different isotypes. The mu and epsilon heavy chain of IgM and IgE lack a hinge region and they have an additional constant region domain CH2-CH2. These domains permit IgM and IgA to interact with light antibodies and form multimeric molecules. Multimeric IgM and IgA also have a polypeptide called the joining or J chain, which is a disulfide link to the constant region and stabilize the multimeric structure. Other constant region domains. The 5 class of immunoglobulin can be expressed either as secretory immunoglobulins SIG or as membrane-bound immunoglobulins MIG. The carboxyl terminal domain of the secretory immunoglobulin differs from the membrane-bound immunoglobulins in both structure and function. Secretory immunoglobulin has a hydrophilic amino acid sequence at the carboxyl terminal end. In membrane-bound immunoglobulin, the carboxyl terminal domain contains 3 regions: an extracellular hydrophilic special sequence of about 26 amino acid residues, a hydrophobic transmembrane sequence, and a short cytoplasmic tail of variable length. Different classes of MIG are expressed at different developmental stages of B cells. The immature pre-B cells expresses only MIGM. Later in maturation, MIGD appears and it is co-expressed with IgM on the surface of the mature naive B cells. MIGM, MIGG, MIGA or MIGE can be expressed by memory B cells. Conclusion. In conclusion, I would like to state that the immunoglobulin is a versatile molecule with great diversity which can interact with billions of antigens that we encounter every day. This diversity or ability to interact with varied antigen resides in its antigen binding site, which is formed by the variable regions of both its heavy and light chains. Within the variable domain of each heavy and light chains, there are CDRs that directly interact with the antigens and give specificity to the antibodies. Their constant region provides extended structure which facilitates its major function of antigen binding. As antibodies can exist as both secreted and membrane-bound forms, their structures are also slightly different to perform both the functions. Thus, the structure of immunoglobulin has many variables and flexibilities to perform all their functions.